gonna spend together the next hour and a half. Our plan is to make a very standard classical proposal just to illustrate you a bit the use of MXCuBE3 and Xray, and introduce the new thing that we have been developing, which is called Braggy. That it's this new image diffraction viewer for visualize the diffraction images while being during the remote operation. So I'm here with Gianluca. Since I get that most of you are familiar with a lot of things that we are gonna show, feel free to interrupt us anytime, or if you have a question, you can use just the Q&A box in Zoom. And since we are two, we are gonna manage and try to address reply either through the chat or directly live according to what is needed.
Daniele, sorry.
Yep.
It's Gerlin speaking. I think you will make your slides available afterwards. Is that so?
Well, in reality, we are recording this, and since we're going to do a live demo, we hope that this can be used and share as a demo for the usage, so that you can also show to your other lab members and show a bit of the software. Yeah.
Great. Thank you very much.
Okay. So I leave the floor to Gianluca. That will start with ISPyB and the sample preparation and the shipment preparation. Yeah.
Hello, everyone. Can you hear me? I suppose so.
Yes.
Great. Hello. So, as Daniele anticipated, we will do a standard, let's say, proposal run, going from the upload of sample names into ISPyB and sending the shipment to the ESRF. Then back to Daniele for the data collection here on site. Basically, it will be the same if you were here physically or from remote. You will see the interface is basically the same. So we start by the simple thing, which is ISPyB and how to prepare an experiment. So I will share my desktop now. Oh, where did it go? Here. Okay, so let me just log out so that we can start from scratch. So usually at home, when you want to use ISPyB, you go to EXI, so the extended ISPyB interface at exi.esrf.fr. We are on MX experiment today, so we just click on MX and I will log in with my proposal name. This is still true and it will be true for a while. And even just to give you a, let's say, sight over the future, even when the data collection will be moved to the individual accounts, at some moment it could happen. We will keep the MX proposal login for legacy proposal, means those who were collected before the change. So you will not have to change on your old data, just keep your MX password safe somewhere. Voila. So now it's loading. Obviously, this is one of our test proposals, so there's a bit more sessions than on average. And you see we have a session today on ID 23.1 where we are sitting now. So first of all, before starting to prepare the actual samples, I will go very briefly through the protein and crystals tab, where we can check a few things. So first, in my proteins, I can see all the protein names, which were synchronized with the user's portal. When you create a shipment from the user, create an experiment from the user portal, and you define a sample in the safety form, we retrieve it from ISPyB and they're all here. You see we have our standard test samples here. We have FAE. We have FDSPs, obviously just me typing random letters sometimes. We have insulin. We should have other crystals somewhere, thermolysin, etc. Funny enough, I don't think I have lysozyme in this proposal. Yeah, I don't, which means I will rename our lysozyme crystal somewhat differently. Here, for example, for the FAE, you see I have crystal forms. If I select, oh, let me increase this, maybe you will see better. If I click here on crystal forms, you see there is one, she's called P212121, where I have the unit cell parameters. I can add a structure, of course, to this. Add a structure means I can add a PDB model. This is already done here. But just to show you, let's just take this second one, which is exactly the same. And I can select different things to add. I can either add a PDB model or the sequence for the protein, the sequence or the UniProt ID. The UniProt and the sequence are particularly important now because if you provide the sequence, we will also try to fetch the EBI AlphaFold database for molecular replacement models, if the protein is available there, of course. So, and other things we can do now here is basically that. And we have another tab over here. So if I go back to protein and crystal, which is ligands, where I can add new ligands. Here, I click on add, and I can add the ligands in different ways. I can upload the PDB structure of the ligand, the CIF, the MOL2 or CIF description for the ligand, or an SDF format containing one or multiple ligands. Now, I'm not creating a new one, but I will show you how then this can be used. So once all your proteins and crystal forms are defined, obviously this is all optional, but it will trigger multiple actions on the auto-processing side. So the more information you give us, the more precise can our auto-processing be. For example, if you provide the sequence, another thing which is very important is that the automatic SAD will run with appropriate solvent content and not just by guessing different solvent contents. This obviously improves the quality of the results. So, but let's suppose this is already on your side. What we want to do now, we have fished our crystals and we want to send them to the ESRF. So I think you're familiar with this. We go to shipments, add new. So we call this shipment user meeting tutorial _1. For the session of today. Obviously, for you, you will see here all the future sessions for your proposal, which are already synced with ISPyB. So this comes from me, and return address is the same from the shipment. One important thing for BioSax, even if you don't want the table back, your box back, it's better to say that the return shipment is the same. We will obviously throw it away, but the generation of the labels is done in the two senses when you upload the shipment. So it's important to also add the return shipment, otherwise you will not be able to generate your shipment labels. Once I'm okay with this, I have no comment to add to this, so I click on save. And I have now, as you see, an empty shipment. Shipments in ISPyB, for those who are not familiar, are categorized in this way. So there's a shipment, which is the full ensemble of samples that you will send. And then they're organized with the same logic as you do physically. So we have parcels. A parcel corresponds to the dry shipper you are sending. Each parcel, so now we will add one. We'll add a parcel which is called UR1. Storage condition, obviously, it's a UR, so nothing special. Voila. They just save. And now you see here inside my shipments, it contains one parcel, but this parcel is empty. It's an empty UR. What I need to do is to put some packs inside. Packs are here on the right, so if you can follow my mouse, here on the right, you see add container. And here I will put, so first my container is a Unipack. And in general, well, this one has a weird name. This one, the one I put in is called ESRF03. It's important that you put the right naming on the packs, otherwise we cannot know what's inside when putting the packs on the beamline. So for example, your packs in general are called CPS 1, 2, 3, 4. So let's just suppose that this is your pack name. I save that. And now you see I have my pack CPS 1, 2, 3, 4, which is an empty Unipack. If I click on the pack again, I click on edit. And now I can add my proteins inside. So I'm looking down because I have notes of what's inside each crystal. So the problem is that, again, in this proposal, I didn't add lysozyme. So lysozyme will be just SD test. Match free. And you see when the crystal form doesn't exist, like for this protein here, for example, I get a crystal form undefined. And then I need a sample name, which and it's very important, but you will be reminded when you prepare your experiment, but the combination of sample name and protein acronym must be unique over your proposal. So let's just call the sample for today, user meeting tutorial _1. And now if I scroll down, this will be named 1, 2, 3. Actually, now I double clicked on the square and it filled the empty spaces. Then let's just upload the other crystal. So I have 3 trypsin crystal here. 1, 2, and 3. And you see when I have a sequence of number, they are pre-filled. This is trypsin and not taumatin. Okay, then I have 2 insulin crystals. 2. And I have 3 taumatin crystal. And they can drag and it will be added to the other ones as well. Don't know why it's defined as P1. And you have 1 thermolysin and 2 FAE crystals, 1 and 2. Okay, so now we are ready to go. So I will save the experiment, but just to show you very briefly here what we can do. So for example, here on the right, you see this is very important because I showed before ligands. I can add one of the descriptor of ligands inside here. This means that if the automatic molecular replacement is successful, then the software will also try to fit the ligands inside. These are the ligands that we defined together earlier. So I'm not putting there because these crystals don't contain ligands. And these columns here in the middle are mostly for when you send crystals on massif. So you will see that in the second tutorial with DDA and MAT. Just to give you an idea, if you click on experiment type, you can select the different MXpress workflows. So now I'm not uploading anything more than this. I will just save, return to the shipment. Okay. Voilà. Actually, I made a typo when I added the experiment, I think, so let's just discard the last modifications. So once I'm done with this, let's imagine I've uploaded also the other packs. Now my viewer only contained 1. I can print the labels by clicking here on the right. You see now I've downloaded the label for my shipment, which looks okay. I can stick those to my viewer and send the shipment to the facility. Need to refresh because I just printed the labels. Now you see the send shipment to facility is active. I can click here. And the viewer has been now, the status has changed and it's now sent to the ESRF. Obviously, it will not pass through the store, so it will never become flagged as at the ESRF. But let's just suppose that time has passed by and the viewer arrived here. So now it's experiment day, and Daniele, who's your local contact, has just put the samples inside the robot. To synchronize the names with MXCuBE, the first thing to do is that we need to tell the software where the crystals are located in the sample changer itself. To do this, we're going to prepare experiment over here. And you see, actually, this is an old one. I removed the old one. Now I will display all our viewers, and you see we have between all the viewers for this proposal, we have our UMT1, which was sent to the SRF. Obviously, it hasn't physically arrived, but they can still put it in process. We can always force a viewer in process in any situation. Now my viewer will be attached to the session on 23.1 today. So if I go to next, you see I see my pack, there's only 1, and the representation of the new plate of the Flex HCD. I think you've been updated on this, but since September, I think, we have removed a few spine pack cells to include more unipack cells. So instead of having 4 and 4, now we have 2 and 6. So, but you don't know, but now I will just put my pack CPS 1234 in the corresponding position inside the viewer, which is position 5, cell 5, position 1. Voila. Now you see the sample changer has a pack inside. I can click on these. Here I will see the name of the pack, the sample changer location, and if I go over, we can see all the names for the different crystals, both the protein acronym and the sample name. These will be used in the data collection step to synchronize the names and create all the appropriate folder for the data collections. So at this moment, we are ready to go in MXCuBE. So I think first I will check if
Gianluca,
we have
may I
questions in the
ask you a
chat. Yeah, Pedro.
question?
Yes.
So you click on once you have the graphics display of the robot, you just click on the pucks and
let me do it again.
It will automatically assign them to the right position.
No. So let me do it again. I will just unload. So I download it from the beamline as well. So first I select this pack here, which is 23.1. Then now you see the pack name is selected because it's blue. Otherwise, it's I don't have another one to show. Okay. Now you see it's white out. I click once, it's selected. Okay. And now if I click on the corresponding position, the pack is now assigned, but I had to click on the position to fill it because the software until now doesn't know where I put the pack. This step is done physically only, and then we synchronize the information this way.
So if you would have more pucks, you would have more lines on the left.
Yeah, obviously. Yeah, let me just show you just to show how it looks like on a normal day. I will select another shipment. Yeah, here I have a shipment with 2 containers. So let's suppose we receive 2 dewards, like here, for example, you see I have 2 dewards received. And now I get 2 samples. So this was from a previous test, is still loaded, the information. So you see now I have 3 packs. So this one is in 5.1, and let's say, well, we will remove later, but let's say this one is in 5.2, and this one is in 5.3. You see, I can assign them this way up to, how many do we have? 18 unipacks.
Okay, thank you.
No problem. We just remove those. Do you have any questions in the chat, Daniele? No, there's no Q&A. I think we'd only have the chat. Your camera is out of focus. Oh, sorry, Carlos, I just read. Maybe it's just dirty. Puoi accendere la luce, forse quello. Ah, the light is open. No. Okay, so I will pass over to Daniele if you don't have other questions. If you have other questions, of course, feel free to type them in the chat and we will go through during the talks. Now, Daniele will go on with the data collection itself on MXCuBE3.
Okay, so let's sign out. So we start from scratch. So you know that when you're to start your experiment, you log in through Google Chrome or to any other browser to this page, which is mxcube3.esrf.fr.
Danielle, may I just, while you're doing that, I believe that you may have, we're seeing a black box in the top right corner of your screen. It may be the Q&A box. We don't see it at the moment because your screen is black. Let's see what happens when, yeah, I think that may have done it. Yeah.
Okay, yeah. So let's move it like that, yeah.
Perfect, thank you.
Yeah, thank you. So then you are prompt to log in on Chrome. Then you have your proposal account. And as Gianluca pointed out, this in the future will change to be a personal account that will link to the A form, so then to the session of the experiment. And then there you are logging on MXCuBE. Okay. Is everyone familiar with the interface, or they are completely new by? I assume that can carry on. I see Ivars. Ivars, are you familiar with the interface? Okay. So the pucks are located in the sample changer. So either you or your local contact, if you are doing a remote experiment, as position your pucks in the sample changer. So then we can just get this sample from the sample changer. And since we detect the presence of the puck, we are getting here in the list only the pucks that are physically present in the dewar, and with a name that are just reflecting their location in the puck. So then we are synchronizing, and you see that we are getting the sample name as the Gianluca has just prepared. Okay, so then at this point, you can do multiple things. You can either mount sample one by one, just right-clicking on the card and doing mount, or I can just add a few of them in the queue and then start mount on the first one. And you see that I'm automatically brought to the data collection tab, and why the sample changer is preparing for the loading. These are, of course, yes, Pedro.
Daniela, is it still necessary to use Chrome only?
No. Frankly speaking, I got users that were using everything from Opera to Safari and everything. So all of them, they work. The only requirement is that it's a recent browser. Yeah. It's just that in terms of the rendering of the interface, here we use Chrome. So some details might look slightly different if using different browser, but there is no limitation. The only thing that we recommend is that when you're working on a small screen or a low resolution one, it's to zoom the browser to the correct size. So I'll give you an example. If I control scroll, you see if I'm that's too much. If I'm on the wrong resolution, you see that some details, they might not be shown correctly because there is no space for such the buttons and like that. So we just recommend to unzoom until you see the whole interface properly displayed. Yeah, of course here we are on a large screen with a high resolution, so that's not such a problem. Okay. So the sample is mounted, and the auto loop centering as center the loop. Puoi chiamare Sasha per dire di non cercare di venire con Guacamole. And you see, and we have a position, which is the position that has been selected for the mounting, for the first centering from the automatic loop centering. So at this point, I'm just zooming in, and then I can do another centering to find the position that I want to center. You see that the centering, it's orange, it means that it's still active. So if I want to improve this centering or select another point, I don't need to reactivate that because this is already active, as the backlight is active. In fact, we see the sample. So you can just zoom in a bit and just re-click anywhere just to reactivate the 3-click centering procedure. So then when I'm happy, I take this point. I can either save it, but save it is not mandatory, because if you add the data collection, the position is going to be automatically saved. Okay. So here then are the list of the parameters for the characterization. One thing I forgot to add is it's a subfolder where you want to store your data, which is something that usually you want to do if you are sharing your bug bin time with other groups. So to do that, I go to setting, and then I make a subdirectory, and I can make as many subdirectory as I want. And this, you will see now, they will show up here. So you're sure that your data from your projects are not are separated from the data from the project of others colleagues from that are using the same time. So at this point, I can either add to queue or run now. Then running now, it just automatically starts the data collection, and with that, we can go to see the diffraction images in Braggy. So same, I'm just gonna go log out. Braggy is this URL here. So it's just called braggy.mxcube3.esrf.fr. So you log in as with your proposal. And you are basically at the same level where your data are going to be written. So data visitor MX1816, which is our proposal. We plan for the future to add some direct link from MXCuBE to Braggy, but this will come only at a later stage. So let's go on 23.1, data of today, row. Made too many subfolders. And then we can look at the image. So on 23.1, since it's a larger detector, loading the images, it takes a little bit longer than on the other beamlines. And this is something that the developers are improving to speed it up. But here you see the first diffraction image. Probably you don't have any. You check it in. Okay, we don't have diffraction, as in every good daemon that we tend to do. Oh no, we have a very faint one. Yeah. So one, there are, I'm gonna show now Braggy. So as you see on the left, we have the sort of tree of the folders where you can see the data. And on Braggy, you can access Braggy whenever you want, not only when it's your MXCuBE session is running. On top, you have the command to adjust the contrast level. You can choose different color scheme, and frankly speaking, they are all very good in order to visualize the images from pixel detector. You can change the scale. One thing that we found that is very good when you have images that are weak is to use this seam log representation. You can add resolution ring, and here on the right side you have the contrast level. You can also add grid, but this is probably less interesting for diffraction measurement. If you want to scroll in, if you want to zoom in, you just scroll on the image, and you see that the images is displayed. And here we see the spots. We don't have a lot of beam today, but you see that for each pixel we see the intensity, we see the resolution of the spots, and then we can go to see the other images. And all the settings that you change in terms of contrast range are preserved when you move to one image to another. So it's not like ADXV that it rescales automatically in most of the cases. And so then you can recompare. And then that. Okay. So you see that just adjusting to match, and you see the diffraction image there. Yeah? Of course, this is intended to provide you with a tool that you can remotely use to look at the images, which is a bit more advanced to what you have from just looking at the JPEG that are stored in EXI, and without the need to use a different image viewer that will have to run through NX or through Guacamole in order to visualize the images. For the time being, we have been focusing on the development more on the performance, so that you can use also remotely, rather than adding other tools, like, for example, line drawings or other more related crystallographic tools. There is also a follow mode, so that if you are in a folder and the new images is collected, it automatically show the new image. Yeah. You have any question?
Hi, Daniel. Is this browser specific?
No.
This is also not, because for the last trip, somehow this did not work for me. So I was wondering if it has any dependable or any browser specification.
No, that should not have any browser specification. But if you let me know on which platform and which browser you were, we can double check. Yeah.
Okay, I'll get in touch with you then later.
Yep.
Yeah. Thank you.
Braggy at ESRF. Yeah, if you have any feedback or questions on Braggy, we have an internal mailing list where you can address your comments and your question, and Gianluca just posted in the chat. Yeah. So once that I have a correct characterization from these images, I can of course go to see the details of the results in ISPyB. So this I'm gonna let to Gianluca do it afterwards. And I can just add the diffraction plan, and this will add the data collection, or will perform the data collection with the parameters that we just got from the diffraction plan. And you see that the transmission is rather high, as said, we are in 35 milliamps, so we are in a rather weak beam. So I can just do it right now, and this will start collecting. And here you see that we have in 35 milliamps, and it explains what is the filling mode and when the next refill will happen. Is there any other specific questions or any specific request? Features in MXCuBE that you want? Yes, so X-ray centering, yeah? Okay. So you see that while this is collecting, the progress bar is advancing. Yes. Can just refresh. Probably the follow mode is not going to perform great on these large images. We still have seen to be a bit of improvement. But you see that these are the images being collected, which is not bad at all. You can just surf through them. Okay, so I guess there was in the chat the request to do X-ray centering, in case of crystals that are hard to center. So that's what we're gonna do. I'm gonna put the light in. And you know that to zoom in and out, there is also a shortcut, which is holding Z on the keyboard and moving the scroll. As for the rotation, if you hold R on the keyboard and you scroll, it will just spin around. Yeah. So let's do a mesh. Let's assume that the crystal is smaller than what we see, or that we want to find the part of the crystal that is better at diffracting. So the philosophy of interacting with the sample is exactly the same. Just do a draw grid. So you see that it's orange, so it's active. And I just draw one. When I'm happy, right click. For the grid, I have to save it. This is something that we might improve in the future, and to work exactly as the point, so that you don't need to save it. But when you add the data collection, is it automatically saved? And so then we do an X-ray centering, or we can do the 2 mesh scans that Sasha has presented you today, or we can do just a mesh scan. The mesh scan, it just does this mesh in 2D, while the X-ray centering, as you know, it does the 2D mesh, then it rotates 90 degrees apart and does a line scan on the best position of the 2D mesh. Usually, if you're not sure if the crystal is diffracting well enough, it's faster to do X-ray centering, because if it detects the well diffracting spot, then you will get also after the line scan the center position. While if there is no satisfactory diffraction in the mesh scan, it will just end after the mesh. Then we do a run now, and I'll be prompt for to confirm the parameter for this X-ray centering. And here in the interface, you see here below, there is a log message that it's updating. So now it's starting the workflow. It asks for to wait a little bit. It's slightly slow. Yes. And here are the parameters. C'è un po' di delay. As in most of the cases, the defaults are what you really want to use. So the usual recommendation is don't change it unless you know what you are doing, basically. So we just submit. And then it's preparing to do the mesh. When the loop is being scanned, you will see that you have the feeling that it's moving quite a bit far away from the position that you draw, and you will see now in a second when it starts to perform the mesh. But this is rather normal, because it needs to move a bit back in order to accelerate and decelerate. So don't get worried if you look at the mesh is much larger than what you draw. It's just that since the data collection is very fast, the motor needs to reach the adequate speed, and for that you need distance to accelerate. So helical data collection, okay. Okay, so yes, yeah. So you see that he has got the best position for the mesh. Now it will rotate 90 degrees apart from there. You see that. And then it will perform a line scan on this position. Here it comes. So all the results of all this is also, you can also inspect them in ISPyB. Again, I'm going to leave to Gianluca to show you that afterwards with the necessary plot. So when it's successful and it found a good diffraction spot, it automatically saves another position. So you see that we have a 0.1 and 0.2, which are the two ones that I saved before. And then this is the 0.3, which is the one that has been selected for the X-ray centering. And now, of course, I can use this point to do whatever this is a new thing. So if, as in this case, the detect that the crystal is bigger than the beam, it suggests you to change the aperture, so to change the beam size to match to the diffracting volume that he has checked. For the moment, we're just gonna refuse that because we don't need that in this particular case. So this point can be used as any other point. So I can either add a data collection, a characterization, or do an energy scan. Okay. So in the case of the helical scan, so there are beamline specific sensible time transmission settings. Less Leila. Usually the default one that are defined from the beamlines are the most convenient one. And in terms of preventing radiation damage, the one that are coming from the strategy are the wiser one. Probably Matthew and Didier are gonna show you a survey that they made on all the different data collection that were performed that compares the resolution of that was estimated from the characterization and the resolution that was obtained after data processing, after collecting automatically data according to the strategy. And this, they align pretty well along a straight line. In most of the cases, I mean, we do recommend not to change the exposure time because the detector are performing well with the exposure time that we provide, but rather play with the transmission and not with the exposure time if you want to overexpose or underexpose your sample. So in order to do a helical scan, of course, you need 2 points. So what you can do, you can select the first one, then you hold Control and select the second one. So let's imagine that I want to do a helical between 0.1 and 0.3. And then you see that if I right click, I get different options that I were getting before from a single point. And then I can do just to add a helical scan. And this not only draws a line, as you hear, and then I can define the parameters that will be used to perform the helical data collection. The good thing is that it's data collection, the helical is going to perform along this vector that you see here, and depends on the order on which you select the point. So you can decide whether to collect from left to right or right to left, basically. Okay. So let's collect this helical scan. Then we just collect a bit more images, and just we do. So we see that while it collects and when it goes. So then next I'm gonna show you something that we call the 2D points, which could be a useful tool that is there since quite some time, but we didn't advertise too much. But could be useful whether you are trying to see where the crystal it is, or try to see if a crystal is well centered, or if what you're looking at is a crystal without having the need to do a mesh scan. Ah, ok, boh. Buona che funziona. Ok. So I remind you also that when you do an X-ray centering, the images are recorded in a dedicated subfolder, or the same for any kind of workflow, really, so that it doesn't pollute your folder where you want to keep the data collection with all the images that are collected. And the mesh scan are recorded here with the prefix mesh, and the line scan at 90 degrees with the prefix line. And so here then you can look at the images. So you see that basically you can do very satisfactory remote session just by using a browser with 3 tabs. Yeah. So now I'm going to show you, as I mentioned before, the 2D points. If you want to remove items from the screen, just because they are hiding your crystal or polluting a bit the interface, you can just select and then hit delete on the keyboard. So 2D points are these. If you right click on anywhere on the crystal, well, actually everywhere on the screen, you have either option. So in this case, I don't have a point, I don't have a mesh, I just right click on somewhere on the screen. So either I can do a go to beam, which is a convenient way to move that bit directly to the beam where I right clicked. I can measure a distance. So if I want to, sorry. So I click on a point and I click on the other point and I get a distance in case I want to measure the size of a crystal, for example, for radiation damage calculation or for publications. And what I can do as well, it's either data collection or a characterization with one image. So if I do that, this is going to add a point that you see that is called 2D point. This, of course, is called 2D point because it's a centering point on the projection, but we are not sure that it's going to be a center point when we rotate the crystal. But as I said, this is good. So this is good enough if you want to do just a small oscillation, or if you want to do a characterization just with one image. Sorry, this default needs to be changed to 1. This is to be fixed, and then you can just run a characterization on on one image. And this could be a good way to check if that crystal that you see over there is diffracting, or if that bits that you see over there, it's really a protein crystal or a salt crystal, without having to collect 4 images or without having to collect the whole mesh. In any case, one thing that we use quite often when we do mesh like for serial crystallography experiment, it's also this feature go to beam, because in that case we can move around a larger support and to put it on the beam. So you see that here the characterization is still yellow because it's still running. It says that characterizing, please wait. So I'm going to show you 2 other things. So you can take a snapshot of the crystal, and then it downloads as a JPEG in Chrome. And you can deteriorate the resolution of your video in case that the network performance is a bit bad. However, one thing to notice is that if you have a smaller screen, as I showed you before, the window, it just organizes itself. You might need, as I showed you before, as in this case, to zoom out a bit. Just to make it, but you see that it's a bit small, but still works okay. Yeah. Yes. Just reset. So if you remember, I was adding more than one sample to the queue. So if you go here, I have this sample on which I've performed all the data collection, and you see all the plot that I would have also in ISPyB when I go here. So I see the quality. If I move down, unfortunately, I cannot display that. But on bottom left, I have the dosor plot, and I have the snapshot of the first lot images and all the parameters that we use for the collection. But I have this second sample, which is in the queue. So if I want to move to the next sample, I can either go here and right-click on it, or I have this sample, which is just here in the queue sample. So what I can do, I can just do either mount here or just click next. So if you're working across different samples, maybe from the same protein, from the same project, you can just click on next, and it will just move to the next sample, unmount this one, and load the next one. Do you have any questions? Mm-hmm. Mm-hmm. So the robot is coming. Yes, probably no sample has been taken from viewer corresponding post lot is set to unknown. Okay, probably there was no sample in there. Okay, so let's do another thing. Yes. So in case you want to load more, as I showed you before, I want to load more than one samples to run it in a automatic or semi-automatic mode. What you can do, you can just select a bunch of them just by click and dragging. And then we can like add, I think it was a question about having the automatic pipeline that I like on my see from the other beamlines this morning. That's basically how you do it. You select the sample. What you can do also, you can filter by protein name over here. And then you can just do rather than express E. We add them to the queue. All the prefix and position, this is going to be taken automatically from the sample name that you gave. We do to the queue and we go to collect. Let me check this. And this is going to collect. So in that case, let's see if there are samples in those positions. So it looks like we picked the sample. So this is going to mount. Yes, Pedro. It's also on our wish list. In the past, there's been some discussion with the safety for privacy reasons, so that you can see the actual who is going in and is going out. It looks that we have overcome that problem, and it's on our to-do list. Yes. So the idea is to either have a small window here, or maybe replace the camera view to display that. Yes. So you see, this is very similar to what I suppose Matthew and Didier are gonna display, show you in the next hour. So while this is running, and if you don't have any other questions for now, I leave the floor to Gianluca, and maybe we try to finish like 10 minutes before. So if there is some other questions, yeah.
Okay, here we are again. So as you've seen, we have done quite a lot of stuff in the last 45 minutes with Daniele, and very convenient way after a very long day of experiment is just to not care much to take notes, because ISPyB is doing that for you. So we go back to Xray and see more or less what you have done over the past hour and a half. So starting from the beginning, we did a characterization. So the characterization, again, we got the diffraction plan on the beamline, you've seen that. But if, let's say, you want to check a bit more of the details of the characterization itself, you look for this entry here, you see it's flagged as characterization. And if you click here on the right on the workflow button, you see we can check the results of the characterization itself. Leia asks if there's a way to do the annealing. Generally speaking, yes, there should be a button in MXCuBE. Is there a button in MXCuBE, Daniele? Beamline actions here is not active on 23.1 because our annealer is broken at the moment. but generally speaking, yes. So, sorry, before answering the question, I just clicked here, which opened me this tab, where I have all the results from the characterization. And yeah, so here we can see much information about everything that the characterization program has done. Eventual kappa angle to realign one crystalline axis to the rotation axis on beamline with a mini kappa available, of course. We can check here the estimation of radiation damage. So the estimate of the half-life dose of the crystal, and many other parameters that are then taken into account by the calculation. Important to note, we didn't put too much attention on this, but you see that he says, best has detected that the crystal can diffract up to 1.5 angstrom, while our detector was, I think, at 2.1. So in this case, it's better to move the detector, do another characterization, and re-perform the analysis. The idea is that the detector tries to analyze everything up to the corner of the detector, of course, but since there's too few reflections over there, the final strategy is actually lower. So you see the strategy was calculated only for 1.6 angstrom in this case, but the crystal can diffract farther than that. So that's something you can have an eye on it, but it's also in the messages in MXCuBE, it's also put in advance. Okay, so Carlos, right? Yeah. No, so Carlos, no, there is no way to download everything at the same time, because yeah, it's too much data to go through HTTP, of course. But so to answer your other question, the process data stays in basically forever. It's just a bit tricky sometimes to access it, but you can ask for support to do that, or you can resync. So the easiest thing is probably if you do use like a syncing script to recover your data. Some auto processing will arrive a bit later, so let it run a bit further more than the end of the session. But if okay, it's true. So Daniele tells, I don't know if you heard him, but for all proposals, data may not be available soon, but they are always available for ISPyB. So if you need a particular file from a particular crystal, you can always download it from ISPyB. They are there forever, but not the full auto-processing folder. Those are live along with the raw data for 3 months on average on disk before being archived to tape. Once the data is on tape, we need to ask to be restored on disk before being available for download. Okay. Oh, the phasing is already done. It's not successful yet. Okay, so this is the data collection we did afterwards. Non è P4. Ho sbagliato l'autoprocessing. FI in P2. Ah, okay. So here it's interesting. Actually, you can see something interesting because this protein has 2 crystal forms. Apparently, one is P2121221 and one is P4. Interesting fact: the characterization, if you define a crystal form, will always force the processing to be in the space group that you specified, because obviously it's only for images, so it's better to, let's say, if you have a previously known solution, it will try to enforce that. The auto-processing, on the other hand, doesn't run on the predefined space group. Actually, it tries that, but it tries also other stuff. And you see here that the best solution is clearly in another space group. So how to check the auto-processing results? I click here on latest data collection result, and I get the results, for example, here for parallel proc. Why is it not loading? Weird. Okay. And you see, if you select a pipeline, normally it should show also for parallel proc, but I think some things are currently running yet. You see, you get all the quality indicators versus resolution. You get different plots, which are quite easy to analyze. And by clicking on the individual files, you can download them. So actually, when they're basic text file, for example, the xds.imp, I can just click and open it directly. For something a bit trickier, like the final multi-record MTZ, I can right-click, save link as, and I will download the file, of course. this is for the data collection. And now this protein contains selenium. We are at 14 kV, so there should be some anomalous phasing, but you see it's not strong enough. So in this case, it detected the anomalous signal inside the crystal, but I think it's still running, so we don't get the results yet. But in this tab phasing, you can see whether an automatic experimental phasing has been performed or not. And there is another similar tab now that takes a bit longer to run, so you will not be able to see the results now, where you can find the eventual automatic molecular replacement results. Then I'm going up with the different things that we did. And here I actually have the workflow, the X-ray centering. So I can take the occasion to answer Eugenio's question, which was how does dosor suggest the crystal size? So if I go here and check the mesh results, you see we have this, I mean, what can be a novelty for some of you, which is this crystal map. The crystal map contains, you see, different images numbered with different number, and each individual number corresponds of a, let's say, region of homogeneous diffraction. So inside, for example, all the images flagged as 1, that's a single monocrystal of, let's say, the crystal, and the other regions are subset, or for example, they can be slightly offset, or if there's a crack inside the crystal or whatever, or a rotation or a different level of quality. And so the most homogeneous region here, for example, was region 1. And as we collected our diffraction images with 30 micron beam, it suggested to use 50 because we can cover this much surface, of course. Not really, Gerlinde. I think Gerard or Clemens will be better to comment on that. We run normally, for what I know, we run all the software with the indication from global phasing. So that's with their default parameters in the recommended way. So if you see that systematically, it's probably better to first let us know, but also we can put you in touch with Clemens and Gerard. Okay. Voila. So then for the line scan out of the X-ray centering, still have the problem with the line scans. Okay, line scans are refusing to load lately. But basically it's the same representation just with the line. One important thing actually when you're evaluating your crystals, you know, in ISPyB you can still not see the actual diffraction image, but you get snapshots. And when you are in the workflow results for the mesh scan, for example, you can get the, this is the image which got the highest score in the dosor analysis for the crystal. See, it's quite nice indeed. Okay, cool. Oh, sorry, I went back to home, but let's go back in. And what did we have then? The helical data collection, which, yeah, when you do a helical data collection, is flagged here as helical, of course. Then the visualization is the same as for a standard data collection, but now the results of the auto processing are still not available. So there's not much more to show actually, rather than when you do the helical, yes, you get the flag of helical here, and then number of images, everything is the same. And you get, yeah, the quality indicators here and eventually the results. Now here at the top, if I refresh the page normally, see there's the MXpress workflow that was running before. We can see why it stopped or not, because it seems like it was going okay. Well, then Didier will show you, but basically you can see that everything has been run automatically up to the characterization at least. And if I go into data collections, oh, it failed something. So the data collection failed at some moment. Either a problem of file writing, we are having a few of these lately, or something different. So yeah, again, we are trying to implement the automated workflow on the beamline, on all the other beamlines. There's a bit of fine tuning that has been done, obviously, over the years on Massif, but again, you can ask when you have an experiment if you're interested in running the beamline in automated mode. It can be ready for when you have beam time. So that's the basics for the results in EXI, actually. So if you need, something which is very important during the experiment, over here on the left, you can change the visualization. So here is the expanded collection list view. You can get what we call the collection summary. So you see these are all the data collections we got, but in a rather more compact way. So we get here our characterization with the results, our data collection with the auto processing, our Xray centering, you just get that it was done, the helical data collection waiting for the auto processing results, etc., etc. And even more useful, I find, is this option here, which is the container view. So here you see the third icon here is container view. And in container view, you see which samples were collected. And you can track this way how many samples you have left, etc., for how many you have a data collection running, and so on and so forth. Yeah. In the meantime, you can see here that MXCuBe is still running, even though there's something stuck, I think, in the workflow engine itself, but yeah, actually, we are aware of that, Adeline. So Adeline says that she remarked that the ugly mole representation, which is the 3D visualization for maps and electron densities after automatic processing, is not working, and that's true. We are working on that. It's not really dependent on us as ISPyB team, something which is above. So we are trying to find a fix for that, but it's not really easy because it's a sneaky problem of file writes that change every now and then. Yeah. Magari ne monto un altro nel frattempo. Yeah, Daniele was pointing out, which is quite useful, is that the dosor plot is a very useful thing. It tells you whether your crystal is badly centered or not. So, for example, in this case, you see the crystal is quite well centered, and the dosor score on the left changes a bit depending on the direction. That's normal, but it's always above 1,000 for all the 4 characterization images. And if you check for the data collection, you see that, again, the score is uniform over the data collection, just drops slightly due to radiation damage, of course. When your crystal is centered not so good, like here, for example, you will get something like this. So a huge variation in intensity by a factor of 2 in this case. Still probably can work, but you see the drop is quite clear. And something which is important to notice is that in general, a drop in the intensity, summed with an increase in number of spots, can be a sign of the presence of ice on your crystals, obviously. This will increase the number of points detected on the image, but it will drop the quality of the crystal overall. Can try maybe to mount another sample, just sì, non è neanche partito. Yeah, that's the problem. Fa vedere qualcos'altro, Daniele? Non penso essere. But I think we have shown you more or less everything we had to show, so I think the next few minutes can be open for questions and then a quick break before you go for the other tutorial. So if there's any other questions / comment or anything specific there that we forgot to show either in MXCuBe or ISPyB. Okay, so if there's nothing, then I think we can thank you all for participating, and yeah, no problem, Adeline. And yeah, and see you hopefully earlier than next year at the users meeting. Don't know, Daniel, if you want to say goodbye as well.
Well, thanks a lot for attending this tutorial. As usual, if you have any question, comments, just drop us an email. We're always happy to hear. You know that we cannot always fix everything or add new features on the fly, but we try to do our best to listen to your needs. At the moment, yeah, we really appreciate your feedback on Braggy if you can use it remotely, since it's the last baby that we rolled out. And well, see you soon.